So in our laboratory, we have sequenced extensively the DNA, the entire mitochondrial genomes, large chunks of the Y chromosome, the X chromosome, and other genes, to work out a very accurate evolutionary history of all of these overall monkeys and apes. Other labs have sequenced the HIV and SIV viruses, and using exactly the same techniques, created an evolutionary history of these viruses, with which we can then overlap with the history of the animals themselves, and can see which animals may have in history transmitted it to which others. And if you carry it long enough, you gradually develop immunity to it. But we're talking hundreds of thousands of years, if not millions, not decades, like humans have been exposed to HIV. And we also know that if you take one species of SIV virus and inject it into another, they get sick, just like we do. So what we have been doing is, first of all, figuring out that the virus has jumped from old world monkeys and apes to humans at least a dozen times in the last 100 or more years. And we know this because we can look at we can go to a village in Cameroon and find the local monkeys in that forest have a specific variant of the SIV virus, and the few local hunters or cooks or other people who have SIV in that region have the very same version of the virus. So the humans in one area have virus that's directly related to the animals directly in that forest. You go a couple 100 kilometers away, the monkeys there or apes there and the humans there have a different version of the virus, but they always match. And so we have been going through and looking at various molecules, either the receptors that allow the virus to dock to our cells and gain entry to them, or the molecules of the immune system. So we, by looking at them across all of these different species of monkeys and apes, we can see the genetic changes that have accumulated over thousands and millions of years, how that changes the shape of the molecule, and that's the kind of information vaccine researchers and drug researchers can try to mimic. Nature has solved, nature has cured AIDS. We now just have to figure out how she did it. And by comparing 20 different species proteins to see the species that are immune to it or not, how those proteins have changed, it gives us suggestions how to make a new protein that might work in a drug or a vaccine. We can also look at different parts of the immune system and try to see how the immune system has evolved. The immune system both is evolving over long periods of time and literally, if you will, within our lives, as all the different variable domains and stuff swap around. So we can, again, try to figure out how these animals, dozens of different experiments, if you will. If you think of every species as a different experiment, this experiment has been repeated 50 or 60 times in nature, and it has almost inevitably come up with the same results. So we see the same protein changes, the same molecular changes occurring again and again and again in evolutionary history when they're exposed to HIV or SIV. Maybe that's going to help us get at this. So we've been looking at, we've sequenced most of the genes of some components of the immune system in dozens of species of primates trying to work on this. We are also working similarly with malaria. There are species and subspecies of monkeys. So one group of monkeys might live in the Philippines, others on the mainland in Indonesia, and one species gets malaria and can die from it. The other species just waves it off and has nothing worse than a bad cold. And so malaria is one of the huge killers of people throughout the world. And again, nature has multiply solved how to cure malaria or how to prevent malaria in our closest living relatives. So using the very same strategy, looking at the monkey's evolutionary history, looking at the parasite's evolutionary history, comparing receptors, immune genes, and so on, we hope to elucidate how these animals have cured, if you will, malaria. Other research I do, as I alluded to before, is what we call conservation genetics. So there's an island off the coast of Equatorial Guinea called Bioko Island that has lots and lots of different monkeys. We don't know how long ago those monkeys got there, how similar they are to the ones on the mainland. If they're so highly endangered, would it be good to bring in a few animals from the larger population on the mainland to breed with the ones on the island? So by looking at how closely related they are, where they came from, how long they've been split, we can try to identify species and populations that national and international laws and treaties can protect, saying that this now goes on the Endangered Species Act, or the Convention in the Trade of Endangered Species, CITES, which is like the international version of the Endangered Species Act. But we're using genetic data to do this. And again, we're using genetic data collected non-invasively, so we don't have to trap animals, knock them out, draw blood, and so on. We can actually just run around the forest with pooper scoopers. I have grad students and students on 4 different continents as we speak, running around with little test tubes full of preservative and little tiny spatulas waiting for monkeys to go. We're doing a really neat project of, we're trying to, Diane Fossey's gorillas, which she studied before she was murdered, and the study of them continued on. It's been going on for nearly 50 years now. She went out at the same time Jane Goodall went out to start her studies. But during the civil wars in Central Africa and East Africa, all of the graves, all of the markers of the graves were knocked over or looted or destroyed. Some of the graves were disturbed. And so we have all of these gorilla skeletons that we can no longer identify back to the individual. So we're doing the equivalent of a forensic study. And we are, because there were samples of some of these gorillas collected 30, 40 years ago, we're able to, like CSI, we're DNA fingerprinting individual bones and tying them back to individual gorillas, to which extensive behavioral data was collected. And then we're looking at growth rates, disease patterns, and other things behind the bone. So we can do some very interesting things by looking at their DNA. I also said that we can try to better interpret the fossil record. Fossils are almost inevitably inherently incomplete. And so it's very hard to build evolutionary trees solely from fossils. But by building very good evolutionary trees using DNA sequences, and using the fact that mutations occur with some regularity, almost like a ticking clock. I like to use the carbon-14 decay. You never know when a particular carbon molecule is going to decay, but you do know over 5,700, 60 years, half of the molecules are going to decay, so-called half-life. We can sort of use genetics like a half-life to try to date when animals last split. And so by looking at the living species today, and then tying them back to closely related fossils that, even if they're partial, we can say they're probably a macaque fossil or